Hello, everyone. This is Suhail Al-Janabi from the ABS Capacity Development Initiative, and I would like to welcome you to the third of our technical webinar series on digital sequence information. This is just to remind us that we are having this technical webinar in the framework of the science policy process, which has been called for at COP14 in Sharm el-Sheikh, where, as we know, divergences and of opinions on DSI were acknowledged, and in the framework of the partnership between Norway and South Africa, the ABS initiative is trying to facilitate that process with webinars as this one, dialogues, studies. We'll get to that at the end of the webinar with a little bit of an outlook. And these webinars in particular are meant to feed into the preparation of the open-ended working group 3 of the post-2020 framework in consultation with the co-chairs and the secretariat of the convention. So yes, this is the third webinar in our series of 3 after having touched upon the role of indigenous peoples and local committees in relation to DSI capacity building, and today, yes, on the commercial applications. Next slide, please. So what is it that we have in mind today? We will have a presentation of the different phases, the 2 phases related to digital sequence information by my colleague Hartmut Meyer. We will have, because that is of course pretty relevant to understanding commercialization questions in relation to DSI, a little snapshot on relevant intellectual property aspects. We'll have a roundtable with experts that are dealing with digital sequence information, be it as a scientific topic or really hands-on and treating with them, and even with a view to commercial applications. And then we will, of course, allow as much as possible some questions and answers with the panel, and having then a wrap-up around 3 o'clock. So I hope that gives you an overview, and with no further ado, I would now like to hand over to, I believe it's Hartmut.
Yes, exactly. Thank you, Zul, for the introduction. And I will introduce now in the topic and show you my slides. Here we are. Yes, I like to thank all of you for attending this online event, and we are again quite a lot of people, and today we are informing about the contribution of DSI in the development of commercial applications. Let me go here. What we like to present is first an analysis on how actually DSI is used in research and development processes, and then also leading to product development, and how these international system of DSI might be connected to the benefit sharing in future. We give a short glimpse on applicable IPR instruments in terms of DSI, and also first results on our analysis of DSI in patent applications. And then we we analyzed a series of commercial products which were produced with DSI, and here I will give 3 examples to show how actually DSI contributes to products, and that then we can also use to stimulate our discussion about how benefit sharing could be done in these cases and in others, of course. So what is apparent, and I think this was also a result of the many studies and the many talks we had in the last more than a year, is that we think that the DSI utilization is actually or can be seen as a process of 2 phases. First, of course, there is the genetic resource from which the DSI is generated under the ABS system, the Nagoya Protocol, the CBD. It is important to have a country of origin, especially when you really access in countries with ABS legislation, then there is a country of origin which in many cases will give you a prior informed consent and agreement, and you also have a ABS contract. And this contract will then set certain conditions for your research and development as the first user. And that contract will also determine the conditions for benefit sharing, which then goes back to this country of origin where you sourced the genetic resource. And of course, like in many cases on biological science, the sequences of DNA or of proteins or both will be elucidated, and also that can be regulated in a contract. And then, which is the usual practice, these sequences are stored in international data banks. So the contract might allow this, but the contract could also not allow publication. So that is, of course, something you need to negotiate. The standard, and I think also something which is usually preferred, is of course publication of these sequences for further use. But we also saw in the discussion first that the mentioning of the country of origin in these data banks is only obligatory since some years. So only a fraction of the DSI which is in these data banks actually states where the genetic resource came from. And second, and also that I think is a very, let's say, useful result of the discussion, the MAT cannot be attached to the DSI in the data bank. So there's no technical possibility to upload the MAT. So whatever is written in the MAT about the use of these sequences is then not available for further users. So that is a problem in the ABS system, in the architecture of the system. And then what we like to call the second phase of DSA, then we enter into a, let's say, even more complex process that, of course, these data banks are open for global access. So essentially everybody in the world could access them and download DSI. And as we also know now is that a lot of users, they don't only download one sequence or two, but they download dozens, hundreds, thousands sequences, complete genomes, etc. So there's a massive download of DSI by single users. And then we see that even if data banks would contain the ABS contracts, it might be very, very difficult or also impossible to follow, let's say, 100 ABS contracts which come with certain DSI that you download. So that's also there's a, let's say, a challenge in that system. And then, of course, further research and development can be done with this DSI. There is, of course, non-commercial use. You can have commercial use. And in the end, on this side of the system, so the post data bank system, there is no benefit sharing undertaken. And essentially also it seems to be very difficult to undertake benefit sharing under the current architecture of the system and the current also international policies. So this is how we, after all this discussion, see how that system works. And this is also where we see the major, let's say, bottlenecks in that system when trying to think about benefit sharing in this DSI system. That's what we like to show you in the beginning, and we are later in the examples. You can see that these two phases, so the direct generation of DSI from a genetic resource, and of course then downloading and using it from data banks, that plays both these processes play a role in development of products. With this, I like to go to the second part, to the IP part, and like to invite Pierre to give a short input on the IP relevant to DSI.
Thank you, Edward, and good day, everyone. I'm not an intellectual property expert, and therefore it's on purpose that I'm presenting this, because intellectual property is an incredibly complicated topic, and we can go on and on and on about it. But for the purposes of our discussions today, which is DSI and commercial applications, I thought it would be useful to reflect a little bit on the various kinds of intellectual property that are relevant or potentially relevant to digital sequence information. And one of the first ones that is just very recently, if you look at this tweet here, which was on the 2nd of July, sorry, the 2nd of June, let's not get ahead of ourselves now, is simply the academic credit and recognition that people who create DSI expect and should, of course, in fairness, get for their work. Especially since many researchers are obliged to deposit their data in data banks as a condition of getting funding or as a condition of having their research published. And there's recently been a pushback, especially from the developing world researchers, about the effort they put into creating sequence information and then not getting any acknowledgement for that. Then there's the second point here, the Creative Commons approaches have been suggested. As far as I know, they're not currently used very much in the DSI discussions. But for example, if you think about the BioBricks initiative, right, where you have these small pieces of sequences that can be used to make molecular machines, so to speak, that essentially operates on a Creative Commons approach, and there might be merit in exploring this further. There's also the form of intellectual property is often not recognized as such, which is the terms and conditions that can be attached to access to data. Terms and conditions have got quite a strong legal provenance. For example, every time you install a piece of software, whether it's free or paid for, you accept the terms and conditions. You don't get a choice to negotiate them. You simply agree. Otherwise, you don't use the software. And there are already databases, with DSI databases that use terms and conditions. For example, JustAid and the GBIF, but for downloads only. Yeah? And so one of the things one can think about is, could these terms and conditions be used to create a form of benefit sharing obligation for people who access databases in general or particular databases? It's another form of intellectual property. I think it's also not necessarily recognized as such, is that there are proprietary curated databases out there available. And that what they do is they essentially clean up the data and make it easier to use for particular purposes. And and address very specific uses of data. And in that way, they're quite analogous to, for example, compound libraries that are used by pharmaceutical researchers, etc. And some of these databases have not only terms and conditions, but actually require a subscription fee for access. And then another form of intellectual property that's quite relevant to the use of DSI is plant breeders' rights. And of course, we all know that, well, those of us who are interested in intellectual property around plant breeding know that there's a big debate going on between plant breeders' rights, which has this provision called the Breeders Exemption, which allows subsequent breeders to use protected varieties of plants for further development and further breeding. Versus the other option that's available in some jurisdictions, which is to patent particular traits of a plant variety through what are called appropriately trait patents. And those often could include the sequences that express a particular trait in the plant. And these are, I mean, the reason these are particularly relevant actually is because of the very useful nature of large collections of DSI in plant breeding, where they enable plant breeders to search for particular traits and particular parent lines, and then really speed up breeding quite significantly. And then there's what a lot of people think about first when they think about intellectual property, which are patents. And now, on patents, there's also quite a few subtleties that are important in the discussions. One of them is collaborative pooling of patents, where everyone puts in their intellectual property and then draws from the pool for a common cause. That's quite common in the pharma industry. There's also this phenomenon of patent thickets, where you so many overlapping patents on in a particular area that it becomes very, very difficult for new entrants to navigate their way through the intellectual property, and effectively blocks further development around certain applications of sequence information. Then there's the question of standard essential patents or patents on tools, where a basic technique that you would need to work with genetic sequences or with genetic resources in general is patented, and you can't use the tool without paying for that, and that in a way hampers, or it restricts who has access to that tool and to those standard essential patents, therefore restricts further development. And then, of course, there are creative approaches around licensing all of these patents. And I think one of the things we haven't discussed enough in the ABS discussions around intellectual property is what use can be made of creative licensing and cross licensing of patents. And then there's the strongest form of intellectual property, probably, which is trade secrets, where a business simply does not disclose the underlying intellectual property, but uses it, in a secretive way, and in fact makes efforts to protect it, because that's a requirement for it to be legally protected. And I think in the DSI arena, the most common use of that is where large commercial users download all of the publicly available sequences, and then use them in-house to evaluate their own proprietary sequences that they have sequenced and own, and that they have information about. And so in that sense, the global commons of DSI serves as a framework for adding value to sequences that are not shared, not made available, and also not published in patents. So it really is very strong protection Where is fair and equitable is another question. And then there's a somewhat different category, which is the traditional knowledge of indigenous people in local communities that is associated with a particular sequence. It's also been referred to as embodied ATK, where the traditional knowledge actually shaped the phenotypical characteristics of a resource. But of course, that knowledge is not itself encoded in the genetic sequence, except to the extent that the genetic sequence is what it is because of centuries or millennia of selection by indigenous people in local communities. And all of these IP considerations are potentially relevant to the commercial application of DSI, and we will come back to them after we've looked at some specific examples. Thank you, and back to you, Hartmut.
Now I'm audible again. Thank you, Pierre, and sorry for changing the slides. I was trying with my computer. Yes, and very quick. This is very recent analysis by Paul Oldham, who's doing a lot of pattern data analysis. And here we see the first set of data of DSI of sequences in first in patent application. So he looked into the first application of patents, and you can see in the first graphic that the number of sequences really rose dramatically, and since the year 2000, so since 20 years, it is only slightly increasing. So there are roughly per year 10,000 sequences mentioned in patent application. And that does not say that, of course, these sequences are patented. In many cases, they are just used to describe new genes, whatever. So we see this later, but there's quite a considerable amount of DSI in patent documents, patent applications. And we can also see who are the main applicants. And here it's interesting to see this mix of public institutions and private companies. So the institution that has the most patents with sequence data is the University of California. Second is Genentech. Then we have the French Centre National de Recherche Scientifique. We have Hoffmann-La Roche, another French one, Novozymes from Denmark, University of Texas, Novartis. So we have a huge mix of private and public, patent applicants, and that also shows that patents as as part of research and development that looks into commercial applications or prepares to be commercialized is done by public institutions. I will leave it with this. There will be much more information available when we have finalized this analysis, and we of course plan to inform in second webinar about really the DSI in patent, and go again into the patent system and see what this can tell us about DSI. With this, I'm going to the next part, which is actually showing some examples. I told you that we looked into several things, and a very interesting example, which stands, I guess, for a lot of other industrial products, is the production of vanillin in recombinant yeast of this Swiss company. The background is, of course, that natural vanilla is a quite high price product. The main vanilla growing regions in this world face various challenges to guarantee a constant supply and quality for those companies who use the natural stuff. But we can also, of course, see that the vast majority of all vanillin is synthetic. So only a small part of the market actually uses the natural vanillin, and in this part of the market now several companies try to introduce vanillin which is not produced by the plant but by various fermentation or recombinant microorganisms. And here we see the case of Evolver, which has a recombinant yeast in production with 5 new genes and enzymes. That is at least what we get from the publication and patents. And we go into this case because there is a lot of public information how these how the DSI was actually used. So this is the general pathway in a microorganism from glucose, the sugar, to aromatic amino acids. And one product here, the one called dihydroshikimic acid, that is actually the starting point for vanillin synthesis. But this yeast doesn't have the necessary enzymes to go from here, from number 1 to number 8. So the idea was, of course, to introduce this biochemical pathway, which is known, into the yeast. And in the end, 5 genes and enzymes had to be introduced to catalyze these various steps. And then, because of course the company did publications on that, you can read it, and there's also patents. There's quite a lot of information on how DSI was used to make this organism. So we looked into that. Is DSI mentioned? Yes. And then we also looked what is the actual contribution to the invention, and which genetic material was used in the end. And I think what is important that we see that DSI in this research and development process and the product is mainly used in 2 ways. One is it is existing DSI from data banks is used to analyze genetic material, to find new genes, to characterize new genes. And also DSI then either produced by the user themselves or also from data banks, DSI is used to synthesize new genes for specific purposes in these microorganisms. And also, because we have the whole discussion in the context of the Nagoya Protocol and the CBD, it was important to see whether we can find the information of the country of origin of the genetic resource, because there's still a lot of discussion whether actually benefit sharing and DSI should follow the bilateral way the Nagoya Protocol is setting up. And I will show you some of our results, not all, that's just too much. So the first step in this new metabolism is done by a specific enzyme, the dihydroshikimate dehydratase. And this, the gene and the new enzyme, which was put into the yeast, was characterized, at least according to the patent, by using 6 existing sequences, protein sequences. So, of course, it could also be characterized by taking many more, but this is at least what the patent tells us. And then when we go to the data banks and to look up the entries, we see that either institutions directly submit the sequence information, or the other case is that there is a certain publication in which the sequence is also published and then put into the data banks. So we have these cases here, and also there is one sequence was just automatically generated by software using existing gene data which are in the bank. So that also can happen, that there is just an automatic analysis of DNA and then prediction how the protein would look like. So what is the information on the organisms which were taken to produce this information? So we have a certain Podospora, that's a fungus, which was used, of course, because there is no further information. There is no information on where the organism was found. It is probable because it was a French institution that the organism also comes from France. The second one is from the same organism, and here this specific strain which was used to for sequencing by those research groups were described in 1952 already. And so it might, also by a French group, it might also be probable that this also came from France, but it's not said. This sequence from another fungus is currently deleted in GeneBank, is not existing any longer. Then we have a sequence from a bacterium, also submitted by Canadian institutions. It might come from Canada. And one information here relates to another bacterium which was used. And there we at least find in the publication that this strain was used. That strain, this BD4 strain, was described by these two Americans in '61, and they wrote that they got it from soil, and it's could also be US. And only for the last sequence, which is also taken from a fungus, there is the information that this was actually taken from Spain. So that's what we found for this one step, and we just could go on with this, but I think it shows you essentially the main patterns how DSI is used to characterize the new genes and enzymes used in this. Just for the second one, because this is then also used later in the final product. Here also we see that the second step, the second protein and DNA is also characterized by using two other sequences. And here they come from the organisms from which these sequences were derived. They have been described in 1977, and it's from soil in the US. And also here we see that the sequence comes from a laboratory organism, Neurospora, which is one of the research organisms since decades. And so there is clear from which strain the sequence was sequenced. But since these organisms are in laboratories since the 1920s or 1930s, origin of them is actually not really relevant any longer. So that's also some pattern we see that, like with Escherichia coli bacteria, you have certain laboratory organisms from which a lot of genetic information is derived. And when we then look into not just into the description of the genes, but how were actually these new genes, these 5 new genes generated, which are now sitting in this yeast, we see also two ways. First, 3 of them were isolated directly from genetic resources, again from this Podospora fungus, 1 from Escherichia coli, which is also a laboratory organism, and from Arabidopsis. The publication does not say where these organisms came from, but that the Arabidopsis, that's a small plant, was property of Evolver. It was their own. And 2 of the genes were synthesized, taking DSI, which is in data banks. So the first one and this refers also to the description I showed you before, was synthesized using this sequence from this soil microorganism as a template, which comes from the US. And here, interestingly, this step number 4, the gene for this step, was synthesized using a human gene sequence as template. And also here, we see a special case, which is quite common, that these gene sequences for a certain, let's say, gene are not only submitted once in the gene bank, but here, for example, this human gene has been submitted by 10 different groups. Also, the Arabidopsis gene, which is down here, has submitted by 4 groups. So also here, well, the attempts, even if all those would tell where the sequence came from, the organism, here we have a problem in, let's say, really following this bilateral approach of the Nagoya Protocol. So that's our conclusion. There were like 25 DNA and protein sequences mentioned in the patent. In 90 cases, this was uploaded more than 10 years ago. In 5 cases, there was information on the country of origin. And as I said, in the end, the publications show that DSI, which is, let's say, present in the product, is either in the form of new genes that were cloned from genetic resources, these 3, and which were characterized by using DSI, or there are genes which are actually synthesized based on taking DSI as templates. And this is what we also know from a lot of research and reports, that it is very likely that much more DSI was used in the whole R&D process. Here, the publications and patents will only show a few examples, and maybe the most relevant examples of DSI which was used. I think this is one example. I'm going to be very quick with 2 more, because we will also share this publication to interested people. Also in the perfume industry, there is use of DSI. There is quite a drive of many companies to produce, let's say, major fragrances, major oils by transgenic organism. In this case, a product called Clearwood, which has a patchouli smell. Also here, there are like 6 genes in transgenic tobacco. I'm just pointing to some interesting stuff. So these 3 genes here are all synthesized by PCR. And the sequences which were used as templates are characterized here. And that's also interesting. For example, this patchouli plant was actually taken from a garden, a nursery in Switzerland. So it wasn't even accessed in country of origin or in situ. Here, the second example is a gene from chicken. It was sequenced from chicken liver, which was probably bought in the shop. So also here, countries of origin are hardly available. And here we again have the Arabidopsis, which interestingly originally came from German collections, but now of course are spread all over the world in laboratories. Yeah, and finally the product which is really produced now is not done in tobacco, but in transgenic yeast. And there's one new gene, and that comes from the valerian. Also this plant from which the new gene was isolated was bought at a seed company, although it's an Indian variety of the valerian. and it was also then screened again against several other genes which were where also the origin is not really clear in the first case. I guess it's just potatoes from the university. Then was screened against a gene from the populace, and this is the huge genome project of the populace. And in the end, this is the strain which obviously now is producing the new scent. Well, with this, I like to close my presentation, and we can proceed with our panel and Pierre. Yeah.
Thank you, Hartmut. Thank you for
Thanks.
really interesting examples there. So now we're trying to do a discussion about this and see if we can share some thoughts with you. Our first panelist today, and I'll introduce them as I call on them. Our first panelist today is Dr. Siva Tambasetti. She's Associate Professor in Intellectual Property Law at the London School of Economics. And Siva, I wonder if you can get us started. As an IP law expert, could you please reflect on the relevance and applicability of my very brief amateur overview of possible intellectual property considerations around the commercial use of DSI? Did I leave anything else out? Did I stimulate any thinking?
Yes, thank you, Pierre. No, it was very interesting to see you list out all the intellectual property rights there. And you also used the word thickets, which I think we can draw on. So generally, when we talk about thickets, we tend to think of thickets in the context of patents and in the context of a company having to deal with a thicket. But a thicket can also be caused by a dense overlap of different kinds of IP rights. And I think that's what you showed so well, that we may not just be talking about one gene sequence, one patent. We may be talking about multiple patents on the same sequence and used in different forms. We may be talking about trade secrets associated with the use of that sequence. We may be talking about private libraries where you have to subscribe to gain access, and of course, the common public databases. So when Hartmut initially put the discussion to me, he sent me 3 questions. So with your permission, Pierre, I would just very quickly go through those 3 questions and then refer to 2 ideas to sort of explain how I see this, both in a zoomed out way and a zoomed in way. So just paraphrasing here, one of the questions that was put to me is it possible to patent gene sequences in their natural form? And would this conflict with the sovereignty of states over genetic resources? This is the nub of the question, right? What is industry's role in DSI utilization with regard to accessing publicly available DSI? And the third question, again, paraphrasing, what can these cases that Hartmut just referred to tell us about the applicability of the bilateral model? So for this, I want to just show you very quickly a diagram that I just put together this morning. So it seems to me that the conversations around DSI and the focus on intellectual property rights is really about trying to either create or maintain a DSI commons. Now, I'm very aware of how problematic the term commons is, and therefore I do use it with purpose. Commons has often been wielded in the context of IP, both as a sword and a shield for and against lower income countries. So I put that there as something that is, that you're trying to create in order to allow the sort of meta-analysis, pursuit of science, these sorts of goals to happen in as streamlined a way as possible. So there are at least 4 things that are characteristics of a commons, right? Anyone can use it. But we know that if you don't have the technical capacity to use it, then a commons doesn't really help those who don't have the capability to use it. The second is that generally from a commons, you shouldn't be allowed to take property rights from it. Now, this is the crux of the question here, and also in the examples that Hartmut showed, if DSI is being used in these patent applications, then to what extent are rights being reappropriated, rights that are freely available in the commons being repackaged and reappropriated? The third point I thought would be relevant is that in order to create a commons, it's very important to be free from prior claims, right? And that's not the case here, because we've got you've got at least two competing sorts of claims on the commons, which is the prior claim of national patrimony or sovereign rights, and the post claim of commercially held IP. Another really important thing about the DSI commons possibility, which I think is perhaps the most difficult to address, is that to create a real DSI, you can't have defection. You can't have one commercial entity reach into the common pot and walk away with property rights, because then that changes the game for everybody. And then you'll start getting defensive patenting or strategic uses of whether you put IP in or don't, or you make this kind of knowledge available. Now, why is it worthwhile thinking about creating a DSI commons? Now, I think we can look to recent history. And I was rereading some of my material about the SNP consortium. Now, the SNP consortium was actually formed by private money. More than 10 pharmaceutical companies came together to invest. I think about 45 million was put into creating this commons. And why? Because creating a commons is a way of spoiling property rights. Because theoretically, if the knowledge or the data is in the commons, then nobody else can reappropriate it. So there is actually a private incentive for commercial interests to create or contribute or even invest in maintaining, creating a commons. Now, that's an idea, I think, that, given money, initiative, and technical capacity, that can really be worked out among those whose interest it is in. And of course, then you'd have to deal with the question of commercially held IP, which in the open source software, again, looking at history, open source software movement was dealt with by license, which Pierre, you do refer to. You refer to the Creative Commons license. The GPL license was very, very concerned, because if one party took the software away and privatized it, then the whole system doesn't work. So this is my sort of zoomed out picture, trying to combine what Hartmut said and what Pierre referred to. Now I want to zoom in a little bit. And I'll be quick here. I'm sorry, I'm so probably going against a number of PowerPoint slide rules here with the amount of text on the slide. But please focus on the colors. So I want to use this slide, which is an example of a marine eukaryotic microorganism, which is patented. And you can get this from Google Patents. You can also get it from Justia Patents, which is both publicly available. To show how homology, bioinformatics, the use of DSI databases aids in propertization. So Thickets. Now I mentioned that Thickets can be just patents, or it can be a dense overlapping of different kinds of IP rights, which is a much more difficult problem to deal with. In this particular case, the invention is this organism that produces lipids and antioxidants and can be used to produce omega oils commercially. So the technology is licensed to a company. I'm not naming the inventor or the company, but this is information that's easily available. Now, there are about 17, as far as I could see, around 17 different patents on this technology that covers different elements of the commercial production or the end product. So I'm often asked the question whether, to go back to the first question I raised, can you patent things in their natural form? So here the marine eukaryotic microorganism is itself patented or exclusively monopolized as one part or one element of the invention. Similarly, nucleic acids that are isolated or characterized from the organism are also patented. Now, this is important in order to understand the degree of alteration that can lead to patentability. It's very low, right? So isolating, purifying, characterizing, which is not technically very onerous, can lead to patentability. Now, this is really important. So when people say, for instance, that, oh, but there's industrial application, even in that case, it's a very low threshold. All you have to show is some use for what you have identified. It could be use that you arrive at bioinformatically. You could speculate on the use. You could disclose an informed guess about the use. All of this is sufficient. The public domain, again, in this patent application, they describe, and I use this example again because it's really quite effectively describe how they use gene bank to identify and characterize the sequences that they have isolated. So this is an example, like Hartmut was talking about, how DSI is used in the characterization of these technologies. So the DSI databases, the availability of that information is important for meta-analysis, but it is also an important and integral element of how DSI is propertized in the first place, right? So it's used to characterize, confirm, identify uses, and establish the scope of a patent application, for instance. Right. So I'll stop the share there. Yeah. So what I've tried to do here is to say, look, there is this bigger sort of political, legal goal of trying to create or maintain this commons. But we have to go through this highly detailed structures that are very difficult to change. And therefore, you can tinker on the inside, or you can propose something that's more radical. I do think there's a lot of purchase in the idea of creating a commons. Again, there is a, we've seen with COVID, for instance, that, those who have the technology can gallop away with it, and those who don't have the technology are always going to be falling behind. So creating this sort of commons or open access model has to bear several very difficult challenges in mind, but I think it's possible to get somewhere.
Right. Thank you very much, Siva. Thank you for reminding us about the important difference between discoveries, which are sometimes not patentable, and creations of the human mind, and also what the thin line is between them at times. And about the commons, let's hope that we can somehow end the tragedy of the commons, which manifests itself as the loss of biodiversity. I'd like to next ask Marcel. Marcel is the Director of Marine Biodiscovery Center at the University of Aberdeen, and he's also the founder of Giox and I think some other companies. So Marcel, as a molecular biologist who has also started companies that use DSI to develop new drugs from marine genetic resources, etc., could you please reflect on how typical or representative the specific examples presented by Hartmut are? And do you have additional perspectives to add on the use of DSI for commercial applications? And of course, feel free to respond to what Siva said and any points that I made. Thank you. Please.
Okay. Well, thank you very much. Again, yeah, it's nice to be introduced as a molecular biologist. My training was in chemistry, but I sort of morphed into molecular biology over my career. Yeah, some interesting points that Shiva raised today. It was really interesting to hear that. And one of the perspectives I'll raise as well from my company experiences is how similar is a sequence to be still usable. Again, it can be a big variation in how much you need to know from an original set of sequences. And the second one I'd like to raise, an interesting point, is that I think that the World Intellectual Property Organization is developing its new standard for digital sequence information. So it's an XML format, standard 26, I believe it's called. That would allow you to directly compare sequences in a patent to those in the database, making traceability much more straightforward if you can decide on the level of similarity that's enough to quote it in a, as something novel. And again, I'll try that as well. If you don't mind, I'll share my screen as well and maybe introduce some material based on my own previous company, not the current company. It's just easier for now to do that. It's at a more advanced stage, that one there.
The more information we have, the more mutual understanding we're likely to arrive at.
So if you can see my screen. So this story starts with a Cyanobacterium, a blue-green alga that appears in many different parts of the world. So again, once you start looking for something, you often find that it's prevalent around the planet. In this case, it was ubiquitous not only in the original Indo-Pacific regions where we started our research, but also in Finnish lakes. And then later on, we found it was prevalent in sequences in other organisms too. But we'll start the story with this blue-green alga. And of course, we got the DNA out of this at a very early stage, did the sequencing, and found the bits of DNA that we needed to generate the proteins of interest that made the molecules of interest that we were interested in. So basically, here's the compounds we were trying to develop as a company. And again, this came out of many years of academic research. So this started in 1996. The foundation of the company was in 2015. So again, it's almost 20 years of work before we get to the stage where it's possible to do anything with it. Now, the important part to remember always, and I make this point repeatedly, is that it's difficult to go back from the chemical structure to the protein that gave rise to it, and to go back from the protein directly to the DNA sequence that was responsible for its production, and it's very difficult to go back from there directly to the organism again. So it makes it very difficult to follow something backwards if you need to. So the way that we worked is, once we had the idea of the kind of enzyme we needed, we would take the original sequence as a query sequence, and I've done it as the protein sequence rather than as the DNA sequence because it's easier to follow and understand. And we would look for analogs. And you can determine that, you know, the lower limit is that those letters of proteins need to be about 30 to 40% similar to have the same function as the protein that you're looking for. So your query protein and your protein that you're looking for don't have to be very similar by other standards. And that's known information. I can point you at the literature that gives you that exact information. But that's very important for setting a limit as to where you say something is similar enough and something is different enough. And again, this is often an argument made in patent examinations as to how similar does it need to be before you can claim it as a novel or inventive step. What happens then is we make modifications to that based on a number of different types of information. Some of them are based on the structure of the protein. So if we have the original structure of the protein here that we derived using X-ray crystallography, that three-dimensional structure allows us to target mutations or changes in the enzyme of interest to make it, in our case, have a better temperature range. So you can use it in industrial processes. You can use it from 20 degrees to 80 degrees or whatever. It might be more stable at a higher pH or lower pH. It might change its specificity towards particular substrates. It might, for instance, be easier to produce in bulk. And of course, for a company, it's important that it's patentable. And again, that's the argument you have with the patent examiner at this point. Often, what we do as well is that we do something called codon optimization, and Hartmut mentioned this. And the key to that is that you're taking each 3 letters of DNA that's responsible for a single amino acid is changed so that it's usable by the bacterium in which you're trying to produce it. That may mean in E. coli, for instance, or in other surrogate host that's used. Now, these enzymes can be used in industrial processes. That's important for multiple reasons. So our industrial process is this one. We're using a synthetic substrate. So this is a chemical substrate made using chemistry. And we add our enzymes to do the process of generating the final product, making molecules that might be useful for drug discovery. Now, the importance of that is for us is that it replaces very toxic, high waste chemical processes that generate a lot of byproducts, a lot of energy is used, and our process is faster. The chemical process may take up to 6 months to do the same thing. Ours takes 2 weeks, so increase of 10 to 20 fold. It's much cleaner. We do it in water with small amounts of enzyme. it is less costly because, again, we don't have to use so many chemicals and treat so much waste. So again, on the positive side, we use DSI, we manipulate it, we change it to make the enzymes do what we want, and then we go from there to a process that will basically allow us to speed up drug discovery to some great extent. So again, this is a series of examples, and hopefully the points I made there regarding the similarity are important. The human inventive steps, using structures, whatever, to modify your proteins, and finally the fact that it does actually benefit all of us in the end by generating chemical processes that are gone from very big industrial processes to what would be in the end a process that is cleaner and greener and cheaper.
So, thank you so much, Marcel. A couple of, very important points that came to me when you were presenting this. One of them is that this is very inventive, and it deserves to be protected as an inventive step. The second one is that it shows the potential of DSI to make global goods and improve everyone's lives. And then a third one I thought was that it shows the danger, or not the danger, but the disruptive potential of this technology. Now, when you disrupt dirty industrial processes, that's great. But when you disrupt small farmer livelihoods, that's maybe not so great. So that would, let me then ask our last panelist, Maria Julia Oliva, who's the Deputy Director and Senior Coordinator for Access and Benefit Sharing and Policy at the Union for Ethical Biotrade. And Julia, I think actually often said this, that you have the best job of everyone in the ABS community, because you're an ABS expert, but you mainly work with ethical companies who actually want to comply with benefit sharing obligations. And even those companies are finding some problems. And so I was wondering if you could share with us some thoughts and perspectives on the ABS complications that are created when a single commercial product uses an input from a range of sources, and a range of sources with different ABS status and IP protection attached to them. And I was wondering if you have any examples of this being resolved through mutually agreed terms or in other creative ways.
Yeah, great. Thank you, Pierre, and thanks so much for the opportunity to be here. I'm very glad you didn't ask me to follow up on Marcel's presentation, because I flunked chemistry in high school, so I was very worried there for a minute. But you asked me about complexity in the field of ABS, and this is indeed something that we do see a lot in practice. And of course, this has to do a lot with the fact that ABS was structured as a system initially where you would have one user and one provider, and we know that this is very rarely the case. I think there's also clearly the complexity that Hartmut has explained, which is that you have this initial phase where you do have access to the resource, but then you can have, all kinds of follow-up uses based on derived products or even information. And I think a third complexity has to do with the issue of scope, which is the fact that many countries around the world now include in their ABS requirement not only research activities, but more broadly just the use of plants and other biological resources in commercial activities, for instance. And I think there the challenge is, and so we do have these complicated cases. So the challenge is, how do you focus? And I think this is key. You said earlier, Pierre, with more information, we can find solutions. But, we also sometimes have to sift through all of this information to figure out where we really need to intervene. And, I don't want to go into too much detail, but the vanilla examples is interesting because there's a lot of information and a lot of things. But yet, for me, if I'm thinking about it as a policymaker, I would say, well, what is the issue here? What is the problem? What do you want to address? Because as Hartmut said, most of the vanillin out there is synthetic and made from quite complex, quite not nice bases, right? So if we can have a better way of producing it, is that a problem? Most of the countries that Hartmut mentioned where vanilla comes from are not where vanilla is native to. And I would argue that the main challenges in the natural vanilla, activities are not linked to research, but to supply chain. They come from very poor places, and there's a lot of problems there at the community. And then the other issue of focus is, as you said, where do you focus? How do you manage, products that have so many biological genetic resources in their composition? And an example that you and I have talked about in the past is fragrances, right? Fragrance compositions include, dozens, often hundreds of different substances, some natural, some synthetic. And there's all kinds of challenges about how you would address such a case. But even without, fragrance compositions can be perhaps a little bit strange, but I thought I would share with you, this is just a very simple hydrating lotion. And it's for men, that's why I've not opened it. But I wanted to show you that even in this very simple lotion, you have so many natural ingredients, from jojoba seed oil to sesame seed oil to beeswax to carnauba wax to different root extracts, xanthan gum, so incredibly complicated. If you have countries that all are requesting ABS processes for the production of this, you're making it almost impossibly. This has to do with scope, but this also has to do, I think, with the need to, instead of focusing on use, focusing on value. And you said, are there any ways in which this is resolved? And the example that came to my mind is actually Brazil. So that Brazil, based on the 2015 law, is basically taking an approach where it wants to track access. So there's a registration system, but not all access leads to benefit sharing. And this is quite innovative in the ABS world. And basically there, you only share benefits if you reach a product, if you reach a final product, and the genetic heritage, as it's called in Brazil, adds significant value to that product. So that would be by the fact that it's an active ingredient, or if it's part of the marketing appeal, which is of course great. To show you another product, I always have products on my desk. This is a lovely facial toner that has roses here. Now, I don't know how important the rose petals really are, but of course it made me want to buy it because it looks great, right? So maybe marketing appeal there is also valuable, even if it's not an active ingredient. So I just think this is a very interesting way to solve this other thicket, to use the word that Theo was using, in terms of really figuring out where is it that there is value of the biodiversity is contributing value, and we can really focus on. So I think that would be one approach that I think could also be looking at some of the DSI examples that Hartmut so well explained so well. I think that could also be important.
Thank you so much. So I'm going to go back to Marcel for a moment. I mean, you showed us how you use sequences from an open access resource that you can basically find in any number of places, and then you can find a relatively different protein sequence that has the same functionality and codes for the same thing. So I was wondering, I mean, in your research and in your business model, and so are there any conditions at all under which you would deliberately use a sequence that has pick and mat attached to it? And what would be the implications of that for your research and for your business model and for your company's regulatory compliance?
Yeah, good question. I'm not sure how to answer that, because what you're describing essentially is a workaround, right? So basically what I could do is I could look for sequences which are from a different jurisdiction where using these sequences is going to be much easier. So I could, for instance, take an American sequence and use that instead. So the difficulty is there a unique case where I would find a DSI on the database that I would have to use for the company, or is there a way to find the same thing elsewhere with less restrictions? And the answer is probably we would take the latter option, simply because it's more straightforward to do that, right? So the problem is, I mean, we have found sequences from something like an Arctic bryozoan, a sea moss, and from an Australian sea squirt that are identical, right? So, I mean, these things are so far apart. And it comes back to the old phrase, which is everything is everywhere, but the environment selects. So it means basically, it's an old microbiology phrase, but it means that basically that organisms can survive in different locations as long as the environment is roughly the same. You'll find the same kind of organisms that do the same kind of function. So it's a really tough question. My feeling is that we're going to have to find a system that pays respect to the fact that these sequences can be found in multiple locations, multiple different origins, not even the same organism. It might be a plant from one location and a microorganism from another location. That adds so much complexity to the question that I can't see anything but a multilateral system working, system that simply looks at making it accessible to the user without restrictions, but then somehow there's a mechanism at the back that says we're all going to contribute to the system in proportion to what we use or what we contribute, or whatever way you can think of it. And it's a different way of thinking about it. I think that's a really important system to try and to look at. I would just like to address the point that you made as well about the small farmers and things like that. So again, there's a classic example there. I think it's the artemisinin example. So there's an artificial process for making the anti-malarial artemisinin based on a yeast modifications to yeast. And again, I believe that behind that, there is a system of fairness that allows the farmers to grow the plants. If there is a failure in the crop, then the artificial system can be switched on. And I would add to that, the importance of the system, the genetically engineered system, is that it's able in future to make derivatives that may well beat resistance. Again, so it's an important, having that available can help with stabilizing supply and making new analogs in the future. So again, there's a positive side to that as well. But not a great answer to your first part of the question, but I think it's the reality is that companies will try and go for the simplest legal aspects. Again, it's just the truth.
We'll find workarounds. I mean, that certainly seems to be the case. I mean, it is also, go back to Siva now, because I see, Siva, that you responded to a question in the Q&A, basically about the DSI commons and what effect countries that don't have access regulations in place will have on the DSI commons. Now, of course, one of the problems of the commons is, as a concept, is that if you allow privatization of the goods, the global goods that are in the commons, then only those that privatize the benefits benefit, and the commons tend to get eroded. Which is, I mean, I see that you've observed here that you know, everyone would need to apply that approach, otherwise you would have enormous leakage, and that has certainly been the experience, for example, in the multilateral system of the international treaty, where users actively avoided material from the system because they could get same or very similar material for free somewhere else. But I was wondering if you have any words of wisdom about how does one overcome this problem? How do you avoid this? As I was saying, when I commented on your first round, how do you avoid this actually driving this really serious sixth extinction, which is really a tragedy of the commons in itself?
So thank you, Pierre, because I realize I may have, because I wanted to be brief, I may have given the impression that I was pushing for a commons. What I wanted to do was to show that we're acting as though it's a commons, but in reality, there are all these other factors that make it not a proper or real commons. Words of wisdom, well, I think with investment, you can engineer commons-like spaces, right? So if you took all of those 4 factors that I put into account, and of course, there'll be much more with greater research that you can find. One important element is, do patent offices take these publicly available sequences seriously as an element of prior art? Now, this is really important, right? Because if you are claiming sequences with certain percentage of similarity to those sequences that are already in the database, then at what point does, as Marcel was trying to put it, at what point does the similarity of a sequence in the database act as prior art and therefore mean that you can't get a patent? Now, that's only going to work if patent offices are completely on board with this and are looking at these publicly available sequences as an element of prior art. Even after 3 or 4 decades of knowing that this exists as prior art, our systems aren't very good at sort of screening for this kind of prior art. So that's a problem you would have to overcome. I think one of the ways it could be done is perhaps to focus on small scale commons that you can create. So I'm making a distinction here between the public domain, a free for all, and more carefully engineered commons. So if Marcel in his particular field feels like there is a blockage and he would like access to certain kinds of data for meta-analysis, then maybe there might be companies, if you're talking about commercial applications, companies that might think, look, the value of precluding property rights on this kind of sequences is greater than the value of allowing individuals to get property rights on particular sequences. So if that epiphany is there, then we could engineer smaller commons. So investment, it's a problem that you can throw money, technology, and political will at. You will need all three, I think.
Yes.
Can I just ask Shiva a question? The question I have is regarding open source software that often is regarded as an example of how a commons can sort of work and how you can get commercial applications based on that. The kind of things I've heard is that, the idea of open source software is you generate some software, you take it from the GitHub, and then you modify it and you put it back on. So commercial companies can construct platforms that are proprietary and they can make a profit from. However, they have to put something back to the main source, or the open source software. And they often say, well, because we are a big company, we have 150 programmers working on this. So 150 programmers worth of time is now being put back into the open source software. Is a model like that feasible for what we're talking about here? So something where you take something, you put something back.
Yeah. So I think that would depend on what was taken and what was put back, right? For something like open source, the way the license works is that there's often a viral element to it. So if you use certain components from that openly available database, then the general understanding is that whatever you produce using that component, to that is also attached the same conditions under which you access that first component. So this would be the strictest version of open source. But yeah, again, this is why I think if you wanted to construct different kinds of commons or different kinds of licenses, then I think that would be possible. And it strikes me as a question for those who are in the technology or who are working there. But what I would not want to see is a way of working around, if you like, this viral element to say, look, I give something in kind or like that over time will make this not function in an open way. I also think, there are many governments, the US government, for instance, is making, taking some initiatives to encourage open source availability of code on federally funded projects. For instance, there's some funding to put this code out openly available, because there is an understanding that the more that's made available, it quickens the pace of discovery and invention. And that's something that we can all draw on. So there will be a sweet spot, I think, for each technological field where there is that element of openness balanced with the element of private initiative.
Right. I mean, I want to invite Julia to comment on that, because, it seems to me that your initial comment about focusing on value is actually a very pertinent one here, right? Where you don't focus so much on the restrictions of the access to the information, but on capturing the value at the end. And would that be a potential solution for, for avoiding workarounds, for creating incentives to avoid the system? If there was it was not a handbrake on innovation and research, but instead a contribution to the maintenance of the commons.
Yeah, absolutely. I think I think it's a real way forward, let's say, which doesn't mean that there aren't a lot of questions to be addressed, such as the one raised with Marcel. by Marcel in terms of the being able to trace back where something comes from, because by the time then that you have it in a final product, how would you have the visibility or the traceability? And that's where the idea in Brazil of a registration system where you are registering each step, that's how they try to get around the issue of traceability, that nobody would, the interesting thing about Brazil is that if you're somewhere in the middle, if you're a company or a scientific institute somewhere in the middle, you have every incentive to comply because it's very easy. All you have to do is register. And by each link then registering, it makes it almost impossible for that last link to get around the fact that this is where it is pointing. But another point I wanted to make around the issue of traceability is this interesting distinction between phase 1 and phase 2, again, that Hartmut was making, because in phase 1, you do have access to the biological sample, and then you have information that you can get from that sample and use in different ways. And that is, as mentioned, really an issue of mutually agreed terms, and making sure that the mutually agreed terms are very solid in that sense. Yet at the same time, and I've just been dealing with a case involving, a negotiation involving this, where the authorities then were calling basically for any information on this sample to be fully covered by the mutually agreed terms. And the problem is that, and it goes to Marcel's point about how some of these substances are, exist in many species in many places around the world. So the company was saying, we feel very uncomfortable saying that all of this now will belong to the government, because we don't even own, this, we can't say this information is out there. This molecule exists in all kinds of places that have nothing to do with this particular project. So that contractual issue, although I guess is beyond the DSI debate, as it's currently happening in the international discussions, is very interesting, and it has to do again with this issue of traceability.
Thank you very much for that. I want to know just quickly, I want to see if we can respond to some of the questions that have been posed by members of the audience, because we are getting towards the end of the webinar now. There's one example, there's one question which has been asked twice or even 3 times in different formats, which is basically, do any of you know of an example where DSI was used and there was actually benefit sharing as a result of it? Or do we have any working examples of DSI use that resulted in benefit sharing? And I suppose one answer to that is that nobody does anything in the biological sciences today that doesn't involve DSI anymore. But do you have any specific examples? Maybe you have some, Hartmut, in your research, or anyone else. Feel free to jump in. Okay, that was a clanger. I take a silence as no, unless somebody speaks up very soon.
There are certainly examples, but it's hard to trace back to whether DSI was actually, where the origin of the DSI was party to the benefit sharing. I've got 3 examples I can show, but they're really hard to show anything actually was shared.
Right. And there was also one question. Sorry, yes, please. Go on.
Sorry, just very briefly, and Pierre, in the study we did in November 2019 for the Secretariat on, sort of scanning how many of the benefit sharing measures include DSI. There are very few countries that explicitly include DSI. So even if there were benefits that came back to the company, we would not be able to tell exactly whether that was because of use of DSI or otherwise. So because the terminology has developed in that interim period, I just think it's not something we can measure or count in the same way. I think we have to assume that genetic resources have been used, and if patents have been filed, then there's some sort of DSI representation of those genetic resources. Whether you call that an element of benefit sharing that can be traced back, that's up for debate.
Right, so that brings me to another set of questions here, which have to do with what needs to be disclosed in patent applications. And of course, and there's also a question that relates to the WIPO IGC work on this. Now, I think I'll answer the first part just to be quick, to save some time. I mean, what you need to disclose in a patent in terms of sequence information is all the information that is necessary for a person ordinarily skilled in the art, I think is the technical term, to work the invention. They should be able to use your patent to replicate the thing that you are patenting. And if the sequence information needs to be disclosed and the origin of that sequence information needs to be disclosed to work the patent, then you have to disclose that. Whether you need to disclose it in what is usually discussed as disclosure of origin, which is specifically tied to ABS, where you're trying to say, I got this from Colombia or Peru or Namibia. Well, that depends a little bit on where you're trying to patent and whether the country's patent laws oblige you to describe the geographic origin or the source of the genetic resources that you're using. And if you patenting in Switzerland, which does require you to disclose origin, there's a question about whether they would ask you for the origin of the sequence if you had not actually used the genetic resource itself. But that also made me think, and I think this is very close to something that you said, Julia, and it's something that I've discussed with Siwa before. In the whole discussion around disclosure of origin in patent applications, right? I mean, one thing that you would need to do to establish a system where you focus on value at the end of the value chain is you would somehow need to say, well, you use DSI in this, right? And I know, Siva, that you did some work around a due diligence provision on, or at least we have discussed the use of due diligence obligation to disclose the use of sequence information. Could you say a few words on that quickly? We don't have a lot of time, but I think an interesting aspect of a potential solution.
Oh, due diligence, or you want me to discuss disclosure of origin?
Well, a due diligence obligation to disclose of DSI as a potential solution, as a trigger for benefit sharing.
Okay, so the reference you're making there to due diligence is from the standard that's in the European regulation that implements the Nagoya Protocol, or at least directs member states to implement the Nagoya Protocol. And what is stated there is a due diligence standard, not of process, but of outcome. So you have to do everything you can in order to have that origin, that information at hand. But this regulation is not linked in any way to IP systems, right? So any due diligence that you exercise as a scientific team or as a research group, there is no route for it to translate into the patent system. So, however, what I would say is, because of the way, in the example that I gave, there are 17 different patents, and each of those patents may also belong to other patent families, which are grouped together. So if one patent in that patent family discloses origin, then there is a multiplier effect for all the patents that are linked in that patent family. So we don't need all countries in the world to have disclosure of origin in their domestic IP laws. All we need is a critical number of them to do so, and this will have an effect around the whole system. So we don't need a treaty. I don't think we need a treaty amendment. We don't need to go under the Patent Cooperation Treaty or other measures. If enough countries start doing this, and I think it's possible because you can use checkpoints, IP officers as checkpoints, enough countries can domestically implement this, and then it will have this sort of systemic multiplier effect. The other thing I would say is that if you read some of these patent applications, I see a great deal of pride when they describe their research, right? So this example I gave you talks about finding this grass in the salty marshes of Nova Scotia in Canada. Now, scientists have this information. They don't disclose this because nobody's asking them to disclose it. And again, this is something Marcel and we've discussed about creating or amplifying an honor system where you go over and beyond the letter of the law, and in the spirit of law and science, you disclose the origin of what you're working with to the extent that's possible. In my view, that would be a better or a more evolved version of due diligence.
Thank you so much. I mean, there's a, well, I mean, Marcel, it looks like you have a comment on that, please.
I would totally agree with Shiva on this whole idea of basically scientific good practice would already say that you should state this bits of information when there is a place to put it. So often you have to go back. If you're doing it right now, you go to a patent, you find a publication. Publication would lead you back to another publication maybe, and that would eventually give you the origin of the collection. And I've even gone back to the 1970s to find things, and it's normally okay. It may not give you the exact location, but give you the country or the reef or whatever you're looking for. So that's not too difficult. It takes a little bit of effort, maybe a few hours sometimes at maximum. But I think what we're looking at now for the future is that if scientists are giving a place to put this information, like with the GenBank, there's a country of origin tag or a location of origin tag, and where that's relevant, you fill it in. Say, I know it came from this country or this GPS coordinates. It doesn't need to be in the patent. We can use either Shiva's system, or the idea is that because the sequences will become searchable quite soon from the patents, you search the sequence, the search sequence from the patent against that in the database, and you'll find matches, and you can probably guess which is the closest match. That will be the one that you're looking for, and that will give you the origin. So again, it's an indirect system. It's not that difficult to imagine that can be automatically linked in future. That would be my take on the technological fix for that, maybe. Right. But again, I think more importantly is essentially the honest system among scientists that says, actually, we would like to give you this information, but there's nowhere to put it right now. So let's make a place for them to put it.
That, I think, it's a very good, I mean, that also, of course, relates to the point I made about academic credit and recognition for people who put in the hard work making the discoveries. And, I know that you're on the borderline between the scientists and the business people. I think that the problem would be that some of your ABS regulators might not necessarily trust the business system, the business community, to adhere to an honest system. Which brings me to a very interesting question that has been asked in the chat about the asymmetries of power, that basically have been shown up by the bilateral approach under the CBD and the Nagoya Protocol. Right? I mean, when we agreed to CBD in '92, there was basically the recognition of sovereignty and the undertaking to provide benefits in exchange for access to the sovereigns. And now we see the shortcomings of that because of the way the DSI is allowing that to be bypassed and so on. So do you have any suggestions, quick thoughts on how this can be addressed? I mean, how this asymmetry can be handled, measures to work with it? Anyone who's first? I suppose capacity development has something to do with it, right? Regardless of what else we do.
I think it's not just capacity development. I think it's I guess you call it inclusive innovation or what have you. The idea is that all actors in a particular piece of research are included right from the very beginning in setting up the aims and objectives of that research. For instance, rather than a company coming in, taking a genetic resource or DSI from that resource and using it for a purpose like heart disease medication, actually let's have a discussion with the people who gave us access to that information and use that to set up a research program that makes sense for everyone.
Right. Yeah. Yes, you will. Please.
Pierre, I'll make a comment on that, but I just also want to point out that Marco de Alessandro has made a very important point about the Swiss origin requirement is not triggered for DSI disclosure. It's there in the comment box. I think Marcel's point is very important, and one of the things I wanted to say to your sort of blue skies thinking question is, I have a lot of hope on scientists and technology. As I think technology moves more quickly than law and political systems, and if we characterize the problem in the right way, I think we can find solutions from the technology. I think part of the problem is that we are struggling to characterize what the problem is. that we want to be solved. And I think that's a big holdup. But if we say, for instance, we want to be able to search gene sequences in patent systems and GenBank, and therefore we need to harmonize platforms, then you can come up with a solution. So I think, characterizing questions in the right way is really important. And I think if we let technology find a solution, it will. But we've got to ask for the right problems to be solved.
Right. I mean, so Marcel's idea that you can use this common standard on sequences in the database to find out where the sequence came from. I mean, from there, it's only a very short step towards training an artificial intelligence to do it all the time and just automatically assign the money to the country of origin. But I don't think we're quite there yet. So I'm aware of the fact that we don't have very much time to wrap up, and that Marcel actually has indicated that he would need to leave a little bit early for a much more important meeting than this one. Thank you very much for being here anyway. And so I want to kind of go for this in a more direct way, right? I mean, when Suha introduced the webinar, he pointed out that we're doing this in the context of helping the science policy process around, the commitment by parties at COP14 to discuss the fact that they have divergences of opinion about benefit sharing for the use of DSI. In my personal opinion, actually, the divergences are mainly around access, but since these two things often get lumped together, even though they don't necessarily always belong together, I was wondering, as some closing remarks, right? I mean, in the proverbial 2 minutes or less, do you think we can find a way to resolve the ABS questions around DSI to the satisfaction of all the different stakeholder groups and sides and points of view involved in the discussion? And if so, how? And Marcel, since you're the one who has to go first, you get to go first.
Yeah, I think it is possible. I think there's been some very interesting thinking so far. So I'd like, the WILDSI project, the Paul Oldham's paper, give us some possibilities for routes by which we might solve this. But I would like to raise another point for the future, because whatever we do, it's got to be fit for the future. And it's already become apparent that current databases already contain non-natural sequences, designer sequences. What do we do with those? And what happens in the next step? And I would like to point to the work of David Baker in Seattle and his Institute for Protein Design. And what's happening there is this group is learning the rules of how proteins are made. So what you're doing is you're taking the DNA, you're taking the sequences of the proteins that are derived from that, and then you're designing something that looks like a protein that exists in nature but doesn't exist in nature. It's completely new. It's a complete, it's artificial intelligence, machine learning, etc., put together to design something that's completely new that does a job that we wanted to do. It might be an enzyme that speeds up an industrial process. Again, like I mentioned, reducing the carbon footprint, solvent, less wastage, less energy uses, etc. What that means is that is completely separate now from the original database from which it came, right? It's very hard to trace that back. It's rules learned from a database. Those rules are then abstracted. Those rules are used to apply to something new. This is essentially, we're still in the observational phase of biology. It's becoming much more the sort of synthetic phase where we can do things that didn't exist before. And then we abstract rules, and those rules can be applied again in the future for something new. So how do we address that problem in the future? Again, so any treaty that comes out of this, any instrument that comes out of this has to deal with that issue. And again, the only way to do that, again, is to recognize that when people put things back into the database, that you can use that, but then somehow, contributions to the database need to be recognized, as well as use of information from the databases needs to be recognized in some way. And again, the only way, in my mind, is a big multilateral system that somehow checks and balances those different contributions and allows benefit sharing around the world.
Thank you so much, Marcel. Please stay around, but if you have to go, then thank you very much for your contributions and participation.
I'll have to go about 5 to 5 to.
Okay, well, then we have lots of time. So, Julia, What do you think? Can we solve this problem, and how can we solve it?
I do think we're, these types of exchanges and some of the, research that is currently being done as to, the different options and the implications is very important. I think a lot of the problems that we run into in the ABS world have to do with, trying to fit reality into a concept, right? So I think in this discussion, we're hopefully avoiding that. Which is, not to say that there isn't quite a ways to go, but I would say what is really important is to try to understand the practical implications and the practical solutions. And also, I would say the specific challenges that we are trying to address. And another important point, and I know I'm not really answering your question, Pierre, but a point that I did want to make here was that let us not forget the other aspects of biodiversity that we're dealing with some of these issues, especially around the issue of synthetic biology, I would say even more than DSI. And we've talked a little bit about, displacing supply chains, but I think there's a real issue to discuss in terms of how do we make sure that biodiversity considerations and livelihood considerations are always a part of decision making by companies moving into biotech ingredients, right? When I talk to companies about biotech ingredients, they tell me things like they're safer, they have a better quality, they have higher efficacy, and also they're more environmentally friendly because they have a lesser CO2 footprint. They come out much better in the life cycle analysis methodologies. And so then it's, I find myself in the position of having to make this point that is increasingly made. You can't look at climate, you can't look at environment without looking at biodiversity. So yes, in some cases you decrease pressure on biodiversity, but you're also decreasing the value, at least the economic value of biodiversity as it stands in a forest. You're also displacing the communities that live from sustainable practices around this. And so for me, a key debate here is how do we bring in these considerations when we talk about commercial use of DSI and synthetic biology. So these are not strictly ABS questions, but I think we cannot find, let's say, a meaningful solution to this issue if we don't also explore these broader topics.
Absolutely, yes, I completely agree with you. I mean, to me, one of the big questions around that is what can you? I mean, a lot of those activities that are being replaced, quite frankly, are not the sort of jobs that you would wish on everyone, right? I mean, they're performed by desperately poor people as a last resort livelihood strategy, right? And if you could create enough value through the use of biotechnology, to instead of forcing those people to walk 20 kilometers and dig holes in the Kalahari in 42 degrees Celsius, right? To rather do something more creative about biodiversity, like planting rare and useful species into that environment and looking after the wildlife population in that environment and boosting biodiversity, through enrichment, that would certainly be a very much better outcome. The problem is, of course, at the moment that the whole system is set up to only pay them for their raw material and then take all the value somewhere else and extract it, not just from the commons, but stop it then from contributing to the conservation of biological diversity. Because, Siva, what do you think? Can we sort this out? I know you've already said we need to be clear about what we're talking about to find a solution, but the conversation has moved on.
Yeah. So Pierre, because I work with law, I think a lot about, whether law is the right way to make some of these changes. And you spoke about power and, things like biodiversity and what we need to do to save species. These are all big questions. And I think we tend to give law more agency than it has when we try to see solutions to these problems from a legal angle. So I think there are both time and language works against us in trying against law in trying to find solutions, because again, science moves far more quickly. I think climate degradation, biodiversity loss is moving far too quickly. We've been talking about this for decades, and there are some comments on the chat box about going around in circles, particularly with the commons idea, with sovereign rights, with the bilateral model. In terms of language, we continue to disagree about basic terms like innovation. What is innovation? Innovation for you might result in harm for me, or an unjust enrichment for you might in effect be a loss on my part. We disagree about things like incentive, right? IP rights are supposed to be incentives, but things like trade secrets or denying technology transfer doesn't fit into any of those incentive models. So when we talk about IP, we have to be careful that we separate the incentive measures from the allocation measures, because IP is also about distributing knowledge and allocating knowledge. So time, language, it doesn't work really very well with law. And there's a third problem, which is because of the complexity of the issues, because of the number of players talking about these issues, because of the different ways in which the issues transversely cut through this complexity, there is a very real impossibility of closure. Even if we end up with institutions and legal arrangements, there are so many different ways of interpreting this that we will eventually end up with a system that can never be the final answer to these problems that we are talking about. So I think one way, now to be pragmatic, I think one way for us to approach this is to separate what we can do now and what is more ambitious thinking. So things like declaration of origin, for instance, that we've spoken about is a very practical thing that can be done now. Improving methods of technology transfer is something that can be done now. The more ambitious questions of creating a DSI commons, how do we make open access models work, that might take more time, that might take different kinds of conversations. And having given this a lot of thought, both in the Nagoya Protocol context and the Oceans Treaty context, with respect to DSI, I think two things. One, I don't see how we can escape a multilateral model that in effect implements a sort of commons-like space. And two, I think any multilateral system will have to work in parallel with the bilateral system. And the hope is the multilateral system will be so good that there'll be less need to resort to the bilateral system. I think that would be a good goal to aim for.
Thank you. That's very, very good. And now, Hartmut, you got the first word, and now you will also have maybe the last one. If you unmute yourself, we'll be able to hear you.
Now you should hear me. Well, yeah, very interesting thoughts, and I can agree to a lot. But I also think that a fundamental topic and decision would be an agreement that products which are using biodiversity, which are based on biodiversity, and that for me also would then include DSI, that from the profit of such products, there should be a contribution to biodiversity conservation. So I think that for me would be a very necessary basic understanding. And if governments could agree on that, then we could come in with all these useful, let's say, ideas about a multilateral system and keeping, like, say, DSI as a common, but with obligations, etc. So I think there need to be movement. Yeah, and if that understanding is reached, I think it will be a huge step forward. And then we could also talk about technicalities. And of course, we look into the system now and see that the system as it is now obviously doesn't bring these benefits, which many people thought it should. Yeah, and there are many reasons for that. And as I as I showed here, of course, with this DSI system, I think we got really stuck with the traditional approaches. Yeah, so there need to be new approaches. Yeah.
Thank you so much, Hartmut. Thank you, Siva. Thank you, Julia. Thank you, Marcel. It was a really interesting discussion. And, you know, I've been discussing this for a long time. When I say it was an interesting discussion, I really mean it. I think we are moving on. It might be slow, but we are making progress. Thank you all very much. Just back to, I think, to Suhal for closing remarks and announcements.
Okay.
Thanks again, everyone.
Thank you. Thanks a lot.
Thank you.
Yes, I can only echo Pierre that this was a really, really interesting webinar, and that we are doing our best now to cut the recording and to put that online on our website for those that want to maybe hear one or the other argument and discussion line again, and then for those who were not able to actually participate in that webinar. I don't know, there is a closing slide, Yannick. I don't know whether you could put that up. Thanks. Because yes, this was the last of our 3 webinars prior to the open-ended working group. But to enrich the discussion even more, there are 2 studies in progress in the final stages that may be also helpful for some background reading prior to the working group. So it is a technical paper on DSI, of course, which looks into the potentials and options and limits of national measures of regulating DSI. So we'll come up with that very soon. We are in the last steps of editing it. We would have loved to present it before that webinar, but it's coming. And so it is with a study on a broader topic, fairness, equity, and efficiency for the CBD and the Nagoya Protocol. And here's an analysis of different cases that use DSI. So that is more on conceptual questions. And we had a pretty conceptual discussion here today. So I think both will be quite helpful. They will be, as you see, on the website of the ABS initiative. Please go and have a look. We are currently relaunching our website. It becomes much more interesting also looking into mobile trade issues, but you're going to be redirected if you still use then these addresses below ABS initiative info topics DSI, for example. Well, all this should feed into maybe one of our flagship events, which is the second global dialogue on digital sequence information, which will be held on invitation and a specific registration process with 120 people approximately in different time zones of the world, starting 28th of June till 13th of July. Based on Chatham House rules. I think for that also this webinar here on specificities of the commercial applications were pretty important, and also some of that more conceptual thinking. And we will do our best to have a wrap-up session on Tuesday, 20th of July, with the outcomes on the global dialogue on DSI, so that it can be then shared with a broader constituency. Really, thanks a lot for your endurance for almost 2 hours webinar with partially more than 130 participants, as I see from really all over the world, from all stakeholder groups, and including also indigenous peoples, business, and regulators, science, and UN organizations. So it may have caused more thinking and reflection, and not provided all the solutions. But I think, as Pierre said, it was really informative, and I would like to thank the participants, the panelists, Pierre for his moderation, and also his input, Hartmut, of course. So thanks a lot